Thank you very much. Now, I hope you'll enjoy this evening. We've got two outstanding speakers, indeed three outstanding speakers. Dr. Bill Hurlbut here, Professor Brian Heap, and Dame Onora O'Neill. Now, we're going to be discussing embryonic stem cells and some of the ethical minefields that have surrounded them. Now, if you think about it, it's no great surprise, perhaps, that it's this area of biology that is now raising the most hackles, raising the most problems. Remember, in the past, it was astronomy and geology that mainly clashed with theological dogmas. And the problem is, I think, and this is a scientist's point of view, that we as scientists are looking at these things from an evidence base, whereas I think it's true to say that certainly in the legal profession and ethicists and sometimes the public in general tend to look at these things from precedent and from fixed positions of philosophy. And what I would suggest is that our understanding, our very great understanding of the worlds that we're living in, is not something that we should deny ourselves. Now, embryonic stem cells, I originally derived them in mice. And these, if you think about all the cells in a body, there are probably about 250 different types of cell, building bricks in our body, each of which has specific structures and functions. And if you just think of it, you've got the transparent cells in your eye, you've got the red cells in your blood, you've got the cells in your muscle. Without these, our bodies couldn't be built. But they all come by a process of sequential specialization from a single cell. I've given the example of building a house using a variety of different bricks, and saying all those bricks come from the brickyard, and just put them into the house. But in the development of an organism, in the development of a human, these cells all derive from the single fertilized egg, which divides and divides, and the progeny then become specialized. Now we know that this specialization tends to be a one-way street, that by and large, one group of specialized cells do not transfer into another. So it's quite interesting to us that it's proved possible to isolate from a very early embryo. This is where you've only got about 100 or so of these cells into the laboratory, which can be maintained and which have also retained that ability to give rise to all the various cells in the embryo and in the adult. And these are the embryonic stem cells. Now, I've just described embryonic stem cells from mice, and the evidence for what I've said is absolutely securely based in experiments in mice. However, it's possible to isolate very similar cells from human embryos, and this is where the difficulties, if you like, arise. It's also, of course, where the great opportunities arise. And it's been seen as a possibility that from cells such as this, it may be possible to generate large numbers of precursor cells that would allow us to repair damaged tissues and organs in the adult body. For instance, to repair damaged heart muscle, or to replace cells in our pancreas that are necessary to produce insulin and therefore help people with diabetes. Many of these are still ideas. However, some of that scenario has actually been tested completely in an animal, in a mouse model. It's not, of course, been tested in human at all. Now, because of this, because of the possibilities, work has been going on with cells grown from human embryos, human embryonic stem cells, and it's raised a lot of worry, a lot of ethical problems. Where we've tried to regulate this, there have been two approaches really, and I would like to suggest that we have the one approach in the UK, where we have gone for regulation by strict licensing under the law, and regulation, if I may say so, Bill, in the US, which I would classify as prohibition, and very much like the prohibition of alcohol. It's totally banned, and yet people with money, not money that comes from federal sources, can nevertheless get on and do what they like. So there's two different approaches to regulation and banning. And of course, what we really should be particularly, I think, interested in is what is the right approach to regulation and what are the right ways forward. And the other thing I'd like to say is that, of course, none of this would have been happening in human embryos if it were not for IVF, if it were not for the ability to culture very early human embryos, fertilize an egg, and culture an embryo in what's usually called the test tube, but it's actually usually the Petri dish. Now, of course, IVF only became a possibility because research was done on human embryos. Nowadays, it's generally regarded as a boon to mankind. It's possible that what we're looking at here is the beginning of a cellular engineering, the ability to change a cell from one state to another for therapeutic beneficial purposes. And it may well be that these cells that we'll engineering these cells will not involve an embryo at all once the methods have been perfected. However, to get there, we probably do need to be using human embryonic stem cells. Now, I don't know if either Bill or Brian, or indeed Onora, agree with me, but that'll at least give you a start for debate. And I'd like now, please, to invite you to present your paper. I should just give you the structure of the evening. Bill Hurlbut is going to give, who is the, I should introduce him, he is a bioethicist, neurologist, neurobiology professor at Stanford, and has been an advisor for the US government as the President's Bioethics Committee, and therefore has come with a very strong background, and indeed I think probably a lot of chips and bruises off you from the whole debate. He's going to start off. Professor Brian Heap is going to come in, followed by Dame Onora O'Neill. So, back in again.
Thank you very much. I want to say what an honor and a privilege it is to be here, and it was so beautiful for one thing, and to share this discussion with such distinguished colleagues. I want to give you a very American perspective, at least the perspective that's driving a lot of American politics, and one that's shared by a great many Americans, depending on how you ask the questions, maybe 50%. And I want to begin with a little insight into the politics of America's discussion on this.
Next summer, I'm going on a camping trip with my friends. On my way home, I'll be in a car accident and I'll be paralyzed for the rest of my life.
In 20 years, I'll have Alzheimer's. I won't recognize my husband or my kids.
Next week, my mommy and Dad are going to find out that I have diabetes.
This is my congressman.
Congressman Don Sherwood.
He voted against federal funding for stem cell research.
Is he a doctor?
Is he a scientist?
Why did Congressman Sherwood bet my life that he knows best?
Help me.
Help me.
Who knows? Maybe I'm your mother.
Maybe I'm your grandson.
Maybe I'm a little girl.
How do you know I'm not you?
Stem cell research could save lives. Maybe yours or your family's. Someone you love. And like Congressman Sherwood said no.
How come he thinks he gets to decide who lives and who dies? Who is he?
Majority Action is responsible for the content of this advertising.
I'm actually not sure what happened to Congressman Sherwood in the recent election, but I would guess he may. So, we're at a crucial moment in the process of scientific discovery. The dramatic advances in molecular biology throughout the 20th century have culminated in the sequencing of the human genome and increasing knowledge of cell physiology and cytology. These studies were accomplished by breaking down organic systems into their component parts, studying them as biochemical processes in the testing for the most part. Now, however, as we move on from genomics to proteomics to discoveries in developmental biology, we've returned to the study of whole living beings. When applied to human biology, this inquiry reopens the most fundamental questions concerning the relationship between the material form and the moral meaning of developing life. The current conflict over embryonic stem cell research is just the first in a series of difficult controversies that will require us to define with clarity and precision the moral boundaries we're seeking to defend. Human-animal hybrids, such as the use of animal eggs in embryo cloning currently discussed in the UK, parthenogenesis, getting embryonic stem cells from activated unfertilized pigs, and projects involving the laboratory production of tissues and organs, these and a wide range of other emerging technologies will continue to challenge our definitions of human life. These are not questions for science alone, but for the full breadth of human wisdom and experience. The scientific arguments for going forward with this research are strong, and I should say I'm a physician. I'm very aware of them. The convergence of these advancing technologies is delivering unprecedented powers for research into the most basic questions in human development. Beyond the obvious benefits of understanding the biological factors in the estimated 150,000 congenital birth defects in the US alone per year, 8 million worldwide they say, it's becoming increasingly evident that certain pathologies that are only manifest later in life are indeed influenced or have their origins in early development. Furthermore, fundamental developmental processes, including the formation and functioning of stem cells and their disordered dynamics, seem to be at work in a range of adult pathologies, including cancer. Yet, from a moral and social perspective, there are serious concerns. This is an 8-cell embryo with the sharp tip of a pin. That's how small what we're talking about is. It's important, however, to acknowledge the many scientific projects for which human embryos could be used, beyond their destruction for the procurement of embryonic stem cells. Some fear the industrial scale production of living human embryos for a wide range of research in natural development, toxicology, and drug testing. Lord Alton, a member of the House of Lords, told me that they estimate here in the UK that there have already been used 100,000 embryos for scientific research. Beyond that, there's a concern about the commodification and commercialization of eggs and embryos, and worry about the implications of ongoing research to create an artificial endometrium, a kind of artificial womb that would allow possibly the extracorporeal gestation of cloned embryos to later stages for the production of more advanced cells, tissues, and organs. Furthermore, from a social and political perspective, the emerging patchwork of policies on the national and international level threatens to create a situation in which there will be commercial motivations for outsourcing ethically controversial and illegal research. Likewise, there will be medical tourism by patients in desperate quests for cures. And in countries like Australia, where embryonic stem cell research is legal but still a matter of controversy, a large number of patients may someday enter the hospital with moral qualms about the research foundations on which their treatments have been developed. What was traditionally the sanctuary of compassionate care at the most vulnerable and sensitive moments of human life is becoming an arena of controversy and conflict. Clearly, both sides in this difficult debate are defending important human goods, and both of these goods are important to all of us. A purely political solution will leave our civilization bitterly divided, eroding the social support and sense of noble purpose that is essential for the public funding of biomedical science. Yet this support is crucial if stem cell science is to advance. In the United States, where there are currently no federally legislated constraints on the use of private funds for this research, there is consensus among scientists and many others that without the National Institutes of Health, namely the federal funding, in support of this research, that progress in this important new realm will be severely constrained. The current conflict in the political arena is damaging to both science, to religion, and to our larger sense of unity as a civilization. The way that this debate is proceeding, at least in the United States, is, in my opinion, completely contrary to the positive pluralism that is the strength of democracy. In May 2005, acknowledging our nation's impasse over embryonic stem cell research, the President's Council on Bioethics, on which I sit, published a white paper that outlines a series of proposals for obtaining pluripotent stem cells, the functional equivalent of embryonic stem cells, without the creation and destruction of human embryos. In the 22 months since the publication of that report, encouraging scientific progress on each of these proposals, we outlined 4, encouraging progress has been reported. As the author of one of these proposals, altered nuclear transfer, I want to draw on this as the foundation for my comments. I will begin by explaining the scientific and ethical arguments that are the basis for moral objection to embryo-destructive research. I will then draw on a discussion of altered nuclear transfer to explore how we might set a moral frame, not just for the procurement of embryonic stem cells, but to open up a broader arena of research in developmental biology. Any evaluation of the moral significance of human life must take into account the full procession of continuity and change that is essential for human development. With the act of conception, a new life is initiated with a distinct genetic endowment that organizes and guides the growth of a unique and unrepeatable human being. The gametes, the sperm and the egg, although alive as cells, are not living beings in the sense of living organisms. They are instrumental organic agents of the parents. The joining of the gametes brings into existence an entirely different kind of entity, a living human organism. With regard to fundamental biological meaning, and I will argue moral significance, the act of fertilization is a leap from zero to everything. In both structure and function, the zygote, the one-celled embryo, and subsequent embryonic stages differ from all other cells or tissues of the body. They contain within themselves the organizing principle for the full development of a human being. The very word organism implies organization, an overarching principle that binds the parts and processes of life into a harmonious whole. As a living being, an organism is an integrated, self-developing, and self-maintaining unity under the governance of an immanent plan. For an embryonic organism, this implies an inherent potency, an engaged and effective potential with a drive in the direction of the mature form. By its very nature, an embryo is a developing being. Its wholeness is defined by both its manifest expression and its latent potential. It is a phase of life in which the whole, as the unified organismal principle of growth, precedes and produces the organic parts. The philosopher Robert Joyce explains, living beings come into existence all at once, and then gradually unfold to themselves and to the world what they already but only incipiently are. To be a human organism is to be a whole living member of the species Homo sapien, with a human present and a human future, evident in the intrinsic potential for the manifestation of the species' typical form. Joyce continues, no living being can become anything other than what it already essentially is. It is this implicit whole, with its inherent potency, that endows the embryo with continuity of human identity from the moment of conception, and therefore, from this perspective, inviolable moral status. To interfere in its development is to transgress upon a life in process. The principle of this analysis applies to any entity that has the same potency as a human embryo produced by natural fertilization, regardless of whether it's a product of IVF, cloning, or some other yet-to-be-invented procedure. Now, the major alternatives to the view that an embryo has an inherent moral status is the assertion that moral status is an accrued or accumulated quality. And this is usually put in terms of some dimension of morphology or function. The three arguments currently given in support of the 14-day limit on embryo research, the lack of differentiation, lack of individuation, and pre-implantation status, these are based in the UK, and actually that influenced the worldwide opinion. These are based on a kind of received tradition that dates back to the 1986 Warnock Commission in the United Kingdom. But this commission explicitly acknowledged the continuous nature of embryonic development, stating, quote, there is no particular part of the developmental process that is more important than any other. In a recent memoir, Mary Warnock discussed the utilitarian grounding of her commission's analysis, acknowledging that her committee's task was to, quote, recommend a policy which might allow the sort of medical and scientific research which was in the public interest. Indeed, I believe that recent advances in embryology do not support this commission's findings. The argument on differentiation is based on the idea that before gastrulation, which begins around the 12th, 14th, 15th day, with the formation of the primitive streak, that blue streak, the idea is that until then, the embryo is an inchoate clump of cells with no actuated drive in the direction of distinct development. It's argued that the undifferentiated quality of the blastocyst at the 4 to 5 day embryo justifies its disaggregation for the procurement of embryonic stem cells, while the evident organization at gastrulation, this is about 21 days, reveals an organismal integrity that endows inviolable moral status to all subsequent stages of embryological development. Scientific evidence, however, supports the opposing argument, that from conception there is an unbroken continuity in the differentiation and organization of the emerging individual. The embryonic axis appears to be influenced by structural organization that may be already established within the zygote, the one-celled stage. Asymmetric distribution of cytoplasmic factors, the yellow in this picture, during the first cell divisions appear to promote different gene expression patterns. And the earliest embryonic cells, at least by the 4-cell stage, appear to be already specified to distinct cell phases. So there's already differentiation occurring very early, at least inclinations, biases. It's not an inchoate clump of cells in any case. All this implies that the changes at gastrulation do not represent a discontinuity of ontological significance, a change in the nature of being, but merely the visibly evident culmination of more subtle developmental processes at the cellular level, molecular level, that are driving in the direction of organismal maturity. These new scientific perspectives were documented in a July 2002 article in Nature magazine. Quote, the mammalian body plan starts being laid down from the moment of conception, a surprising shift in embryological thinking. A second argument is the argument from twinning. The idea here is that as long as the embryo is capable of giving rise to a twin, it cannot be considered to have the moral standing of an individual. Yet, monozygotic twin, that's identical twin, which is what we're talking about here, occurs in just 1 in 240 births, and it does not appear to be an intrinsic drive or a random process within embryogenesis. It may occur much more frequently, and the twin doesn't survive, by the way, but it does not appear that the majority twin. Rather, this kind of twinning appears to be a result of a disruption of normal development, either by mechanical or biochemical disturbances in the fragile cell relationship. Evidence for this comes from in vitro fertilization, where depending on when you implant the IVF embryo, you get a 2 to 10-fold increase in monozygotic twinning, clearly the result of some kind of disruption or biochemical difference. Once this disruption occurs, it seems to provoke a compensatory repair, but with the restitution of integrity within 2 distinct trajectories of embryological development. In considering the implications of twinning for individuation, one might better ask the question from the opposite perspective. What is it that keeps each of the cells of the early embryo from becoming a full embryo? Clearly, crucial relational dynamics of position and intercellular communication are already at work establishing the unified pattern of the emerging individual. From this perspective, twinning is not evidence of the absence of an individual, but of an extraordinary power of compensatory repair that reflects more fully the potency of the individual drive to fullness of form, even at the earliest stages of embryonic human life. Some have argued that the implantation of the embryo within the uterine lining of the mother constitutes a moment of altered moral status. The embryo, however, floats down the fallopian tubes into the uterus and implants around the 6th to 7th day. So if anything, it ought to be earlier if you'll draw that line. But all along the way, the journey through the fallopian tubes before implantation involves diffusion of essential nutrients and growth factors, and this sustains the life and nourishes the growth of the developing embryo. Implantation and the development of the placenta simply extends this relationship between mother and embryo with an internal circulation of the embryo as it gets too large to be nourished by direct diffusion. Us non-placental animals never, of course, develop this. Implantation, then, must be viewed as just another step in a continuum of ongoing, intimate dependence all along the trajectory of natural development that begins with conception and continues with infancy. This continuity implies no meaningful moral marker at implantation. Now, most other arguments relate in some way to the onset of a specific function or capacity. Arguments for changing moral status based on function, however, are the most difficult to both refute and to defend. The first and most obvious problem is that the essential function and even their minimal criteria and age of onset are diverse and arbitrarily assigned. Generally, they relate to the onset of sentience, awareness of pain, or some apparently unique human cognitive capacity like consciousness. This approach, however, raises a number of disturbing ethical questions. So, if human moral worth is based on actual manifest functions, then does more of that function give an individual life a higher moral meaning, moral value? And what are we to make of the parallel functions in animals that routinely are sacrificed for food and medical research? Furthermore, what becomes of human moral status with the degeneration or disappearance of such functions? While we might argue that our relational obligations change along with changes in functions, such as occur with senile dementia, we wouldn't spend the same amount of money caring for this person as we would a 3-year-old. Still, we would not sanction a utilitarian calculus and the purely instrumental use of such persons, no matter how promising the medical benefits might be. More fundamentally, from a scientific perspective, there is no meaningful moment that one can definitively designate the biological origins of a human characteristic such as consciousness. The human being is an inseparable psychophysical unity. Our thinking is in and through our bodily being, and thus the roots of our consciousness reach deep into development. The earliest stages of human life serve as the indispensable and enduring foundations for the freedom and self-awareness that reach their fullest expression in the adult form. With respect to fundamental moral status, therefore, the human being is an embodied being, whose intrinsic dignity is inseparable from its full procession of life, and always present in its varied stages. This conclusion is consistent with 2,500 years of medical science, As recently as 1948, the physician's oath in the Declaration of Geneva, echoing the enduring traditions of Hippocratic medicine, proclaimed, quote, "I will maintain the utmost respect for human life from the time of conception." Now, the principles that I've outlined are the foundation for the prohibitions against the use of federal funds for embryo-destructive research in the United States. Contrary to general impression, this is not a policy imposed by President Bush, but it is the long-standing policy established by the U.S. Congress over a decade ago. It is within the frame of this legislative constraint that the President's Council proposals to solve our nation's impasse over embryonic stem cells, were put forward. Throughout the Council report, there's a distinction drawn between totipotency, the capacity to give rise to the whole organism as an integrated living being, and pluripotency, the capacity to give rise to the many different individual cell types that Professor Evans spoke of. A naturally fertilized egg, the one-cell embryo, is totipotent. Embryonic stem cells are merely pluripotent. They lack the immanent powers for self-organization and self-development that characterize an embryonic organism. As outlined in the Council report, altered nuclear transfer creates laboratory constructs that never rise to the level of totipotency. Drawing on a subset of organic powers, the powers of natural development, altered nuclear transfer proposes the production of biological artifacts capable of forming pluripotent stem cells, but without the creation and destruction of human embryos. This proposal shifts the ethical debate from the question of when, during development, a normal embryo is a human being with moral worth, to the more fundamental question of what component parts and organized structure constitute the minimal criteria for considering an entity to be a living human organism. The conceptual challenge of this approach is to define the boundary between mere cellular growth lacking organized form and a coherent living organism. The scientific challenge is to identify the precise alteration that allows the production of pluripotent cells that are the full functional equivalent of embryonic stem cells, but without the creation and destruction of a human embryo. Altered nuclear transfer is a broad concept with many possible approaches. It employs the technique of nuclear transfer, but with a preemptive genetic or epigenetic alteration to produce a cellular system that lacks the integrated unity that characterizes a living being. In standard nuclear transfer, you all know this probably, but let me go over it. In standard nuclear transfer, the nucleus of a somatic cell is inserted into an enucleated egg. The egg, the oocyte, then has a full genetic complement, and after it is electrically or chemically stimulated, starts to divide like a naturally fertilized egg, at least in some cases. This is how Dolly the sheep was produced. If Dolly was a sheep, then Dolly was an embryo, presumably. In altered nuclear transfer, the somatic cell nucleus or egg cytoplasm, or both, are first altered before the somatic cell nucleus is transferred into the oocyte. These alterations preclude the coordinated organization and developmental potential that are the defining nature of an embryonic organism, but they still allow the generation of pluripotent stem cells. It's a little bit like creating a model airplane kit without the glue. You have parts but not a coherent whole. There's a natural precedent for such a project. Benign ovarian teratomas, dermoid cysts, are germ cell tumors that produce primary embryonic stem cell types, and they also produce more advanced cells, tissues, and organs, hair, fingernails, teeth. It's a bit of a grotesque tumor, but a fascinating one. It's like a bag of jumbled puzzle parts that have unorganized growth, lack the integrated unity of a coherent, organized organism, but still have the power to produce the cells that we're looking for. The exact cause of the teratoma's failure formation is not known, but evidence from animal studies suggests that it relates to one or a few epigenetic changes established in the oocyte's development during gametogenesis. When an egg is activated without a sperm, its imprinting pattern lacks the complementary contribution that's delivered by the male gamete, and this renders the egg inadequately constituted to direct the integrated development of human embryogenesis. The trophectoderm, which is the outer layer of like the soccer ball of the embryo, the trophectoderm and its lineages simply fail to develop properly. Nonetheless, the pluripotent stem cells are formed in the inner cell mass. It's important to note that neither religious tradition nor medical science have ever considered teratomas to have the moral status of a human embryo. This natural example shows how even a small change in genetic or epigenetic configuration can affect the entire balance of an enormous network of biochemical processes necessary for embryogenesis. A functional organism is a dynamic whole, an integrated web of interdependent processes that express emergent properties not apparent in the biochemical parts. Within this dynamic, self-maintaining system is the very principle of life, the organizing information that establishes and maintains the coordinated coherence of a living system. But if you take out certain crucial components, like in geodesic domes, we have a lot of those in California for the hippies. If you take out a component, it collapses because every part is crucial of the crucial parts to sustain the whole system. With a full complement of essential elements, an organismal system subsumes and sustains the parts. It exerts a downward causation that binds and balances the parts into a patterned program of integrated growth and development. Partial organic subsystems, cells, tissues, and organs, that are components of this larger whole, if separated or separately produced, may, however, temporarily proceed forward in development and in function, but without the coherent coordination and robust self-regulation of the full organism, these partial subsystems are in reality nothing more than fragmentary cellular structures whose trajectory of growth is distinct from that of embryos. The underlying idea of altered nuclear transfer is that small but precisely selected alterations performed prior to nuclear transfer. So it's preemptive. These alterations will allow the harnessing of such partial developmental subsystems to produce pluripotent stem cells, even apart from the living organismal whole that is the natural source of these cells. One variation of altered nuclear transfer involves a gene called CDX2. It involves the deletion or silencing of this gene, which is a very fundamental gene in natural development. As described in a January 2006 paper in the journal Nature, MIT stem cell biologist Rudolf Janisch, through a series of mouse model experiments, established the scientific feasibility of this approach. Using RNA interference, he was able to reversibly silence the functional expression of the gene CDX2 in the somatic cell nucleus prior to its placement into the enucleated egg. Without the expression of CDX2, there's a fundamental failure of formation. The first differentiation into distinct cell lineages does not occur, the body axes do not form, and the basic body plan is never established. It's a more fundamental disruption even than occurs in a teratoma. Yet from these laboratory constructs, lacking the character of an organism, Janisch procured fully functional pluripotent stem cells. More recently, studies suggest that it may be possible to achieve the same result through the preemptive silencing of CDX2 messenger RNA in the egg, in this case too before the act of nuclear transfer. University of Missouri biologist Michael Roberts reports that in natural mouse development, maternally derived messenger RNA for this gene, CDX2, is present in the egg and appears to be asymmetrically distributed in the first cell division after fertilization. The asymmetric distribution of the products of this gene, the messenger RNA, may direct the two-cell stage to initiate distinct lineages. Now this paper, it has some problems with it, and it's been challenged and it may be recalled, so I have to be a little cautious in saying this. But he has replicated these results, and they're very, this, what I'm telling you, has further foundation. So it looks pretty good. In the mouse studies, by selectively silencing CDX2 in a single blastomere, a single cell after the two-cell division, Roberts was able to produce an unorganized mass composed exclusively of cells with the character of those of inner cell mass. In other words, just the embryonic stem cell precursors. Furthermore, in a recent presentation to the President's Council on Bioethics, Hans Schuler, Director of Cell and Developmental Biology at the Max Planck Institute in Germany, discussed similar mouse studies in which he silenced CDX2 in the pronuclear stage just after fertilization but before division. He described the product of this procedure as, quote, a single lineage tissue culture, lacking the organization essential to be designated an embryo. Nonetheless, he was able to produce pluripotent stem cell lines from these cellular systems at an earlier stage, the 8-cell stage, and at a rate 50% higher than they can even be obtained from IVF embryos. These research results are consistent with the moral claim that altered nuclear transfer is able to produce pluripotent stem cells from a cellular system that is not reasonably considered an embryonic organism. The preemptive nature of these alterations, before combining the egg and the donor cell, assures that no embryo ever comes into existence. That's the key to this project. These alterations are at such a primary level of constitution that they are rightly recognized as an insufficiency, not a defect or deficiency in an existing being. There are parts, but no unified whole, and indeed no capacity or internally active disposition to form an integrated whole. The absence of functional CDX2 precludes the coordinated coherence and developmental potential that are the defining characteristics of embryonic organization of an organism. In testimony before a Senate subcommittee on stem cell research, Rudolf Janisch stated, because the altered nuclear transfer product lacks essential properties of the fertilized embryo, it is not justified to call it an embryo. CDX2 silencing is just one of a range of possible approaches in which altered nuclear transfer could produce a non-embryonic cellular system capable of generating these cells. If these limited biological constructs were produced only for serious scientific purposes and were accorded the respect appropriate to all human tissues, this project would not compromise any fundamental moral principles. Of course, animal studies with non-human primates need to be conducted before we proceed to human cells. Just a couple of concluding comments. Altered nuclear transfer, in its many variations, could provide a uniquely flexible tool and has many positive advantages that would help advance stem cell research. And indeed, it might be a transitional technology to get us to some of the places that Professor Evans was mentioning. Unlike the use of embryos from IVF clinics, altered nuclear transfer would produce an unlimited range of genetic types for the study of disease, drug testing, and possibly for therapeutically useful cells. By allowing controlled and reproducible experiments, altered nuclear transfer would provide a valuable tool for a range of research, including studies of gene expression, imprinting, and intercellular communication. For this research, it will be essential to study the simplest cell-cell interactions, most specifically those in the first few days of embryogenesis. Once a morally acceptable form of altered nuclear transfer is established, it will provide an effective tool to probe and explore organic processes in early development, even beyond the procurement of embryonic stem cells. So it sets a precedent for all types, range of embryonic type explorations. Furthermore, the basic research necessary to establish altered nuclear transfer would advance our understanding of developmental biology and might serve as a bridge to transcendent technologies such as direct reprogramming. Moreover, as a direct laboratory technique, altered nuclear transfer would unburden embryonic stem cell research from the additional ethical concerns of the leftover IVF embryos, including the attendant clinical and legal complexities in this realm of great personal and social sensitivity. The one remaining link with in vitro fertilization, the procurement of oocytes, of eggs, is a subject of intense research and quite strong moral objection in the United States. But I believe that there are hopeful ways of obtaining eggs without the morally dubious and expensive hormonally induced superovulation of female patients. These include the use of eggs left over from IVF, the in vitro maturation of these eggs, possibly the cultivation of eggs from ovaries removed from surgery and from cadavers, and maybe one day the direct production of eggs from embryonic stem cells themselves. So, we're at a critical moment in the progress of science and civilization. Advances in biology have delivered new powers with extraordinary potential for positive applications in both basic research and clinical medicine. Yet at the same time, these new possibilities challenge the most fundamental moral principles on which our civilization is based. English author G.K. Chesterton has a metaphor that may inform our current situation. Little boys are playing soccer in a field, but right at the edges of the field, cliffs go down 1,000 feet to the crashing waves below. So where are the little boys playing soccer in the middle 20 yards? Nobody wants to do a corner kick, right? And then somebody comes and builds a strong fence right at the borders. Now they can play right to the edges. Our science is something like this. If we could define with clarity and precision what the moral principles we're trying to defend really are, then our science could expand its realm of inquiry. That would be a very good thing. Moral analysis that underlies this proposal for altered nuclear transfer begins with the reaffirmation of the inviolability of human life across all of its stages, from conception to natural death. By grounding this moral valuation in the continuity of organismal existence, we can then draw on the distinction between the full totipotency of a living being and the mere pluripotency of an organic system with fragmentary cellular growth. The conceptual shift essential for the practical acceptance of this proposal is based on the acknowledgement of the moral neutrality of laboratory constructs with only partial and unorganized developmental potential. As a society, we've been through something like this before, when we dealt with the whole question of human parts separated from their organismic wholes, when we dealt first with blood transfusions and then with kidney and later heart transplants. This conceptual transition, however, will be more difficult because our natural moral sentiments equate the dynamics of growth and development with the powers and potentials of living beings. Nonetheless, in the age of developmental biology, we will come to understand that a biological artifact similar in character to a teratoma does not have the moral status of a human being, even though it can undergo organic processes that produce human pluripotent stem cells. As we enter the coming era of rapid advance in biotechnology, the search for alternative sources of human pluripotent stem cells established a positive precedent for maintaining constructive ethical dialogue and for encouraging creative use of our scientific knowledge. In recognizing the important values being defended by both sides of our difficult debate over embryonic stem cell research, this approach could at once sustain social consensus and open hopeful prospects for scientific advance. That would be a triumph for our society as a whole. Thank you.
Bill, thank you very much for a really wonderful and informative talk. I think steering between all the both moral and scientific pillars and privileges. You're getting there, so that's very good. Now, we're going to have a slight pause while we just change over the laptop. One thing I might throw you is that certainly in mouse experiments, one of the very best ways of making a teratoma is to transplant a blastocyst into the wrong place.
Yes.
So what we're looking at in the teratoma is a deliberately misformed embryo.
Yeah, there it's a matter, however, of not having the right environment, not an intrinsic capacity. And my project would create something with an intrinsic insufficiency.
Absolutely.
So
yes.
it's a difference, but that's true. And we have to be very careful before we conclude that what is produced is not an embryo for that reason.
Okay. Now over to Professor Bradley.
Thank you very much, and in the interest of time, I shall try to be brief. Walter Bagehot, the English economist and essayist of the 19th century, wrote, "One of the greatest pains to human nature is the pain of a new idea." And our distinguished chairman, Martin Evans, and his colleagues have certainly a lot to answer for, because they certainly confirmed this aphorism with the first demonstration some 25 years ago now, that embryonic stem cells can be isolated and grown in the laboratory indefinitely. It was a discovery that is both exciting and challenging, because it opens doors, as we've heard, towards the possibility of new medical treatments for serious diseases and injury. And Bill Hurlbut has shown us how embryonic stem cells have reignited the debate, particularly in the USA, about the moral status of the human embryo, attracting the involvement of high-profile celebrities such as the late Christopher Reeve, Michael J. Fox, and Julie Roberts, as well as the attention of politicians and religious leaders. In contrast, in the UK, the embryo status issue was settled in law, at least, with the landmark Human Fertilisation and Embryology Act of 1990 that allowed research on human embryos up to day 14 for problems of infertility. It was followed by the Human Fertilisation and Embryology Regulations of 2001, which provided for research on embryos that related to the treatment of disease, not just fertility, but to diseases themselves. Licenses were granted to create embryos and to create embryonic stem cell lines from human embryos, which had originally been made for IVF treatment to increase knowledge about the treatment of serious diseases. And applications have been submitted, as I'm sure we all know recently, to further such research by the transfer of a nucleus from a human cell into an empty egg of another species. Now clearly there are, as we've heard, serious deontological issues which remain and they continue, and it's evident from the scheduling of this discussion in a series of lectures on science and religion. They include, if I have a duty to heal the suffering of others, how should I judge things or persons that seek to prevent that moral action? Do the good aims of research justify the idea itself? Does the new biology change our understanding of the moral status of the human embryo? So, how did we arrive at our current position in the UK, very briefly? Now, the religious position about the status of the human embryo is mixed, not least in Christianity. The picture we have, for some reason I've lost a slide, but the point I was going to make about Christianity is that certainly among our Roman Catholic friends and Southern Baptists, it would be associated with day 0, the point of fertilization. In connection with our Jewish friends and Muslim friends, it will be much later, 40 days or even later. And with Hindus, it's very indeterminate because of the view of continuity of life and of reincarnation. Now in the Bible, Job in the Old Testament speaks of a caring and loving God at work, forming the human being from the outset. Speaking through Jeremiah, the Lord says, "Before I formed you in the womb, I knew you." The psalmist reiterates a similar idea, saying, "My frame was not hidden from you when I was made in the secret place. When I was woven together in the depths of the earth, your eyes saw my unformed body." Now, some theologians would argue that to use verses in the Old Testament to inform our debate about the unacceptability of ending the life of a very early embryo in order to extract stem cells for research and or therapy is an extrapolation to situations for which they were never intended. The Bible is not a scientific text. It doesn't give lessons in the science of embryology, nor does it define the precise time of the beginning of life. Nonetheless, a lack of specific biblical texts or an absence in the teachings of Jesus does not necessarily imply permission. In church history, Thomas Aquinas, the outstanding philosopher and theologian of the 13th century, adopted Aristotle's ideas, believing that an embodied soul with a complete human nature was the result of the creative act of God over a period of 40 and 90 days for males and females, respectively. Aristotle saw a 40-day period as a minimum for an embryo to become a human being, the soul being the form of the body, or the life principle, that enabled matter to become a human in actuality, an account that remained more or less unchallenged for about 2,000 years. The destruction of the human embryo was generally regarded by the earliest Christian witnesses and the churches of the East as homicide. Hence, the current position of the Roman Catholic Church, enunciated by Pope Pius IX in 1869, that life must be respected and protected absolutely from the moment of conception. And as the Pontifical Academy of Life said in 2003, human embryos are certainly not subjects who can give their personal consent to experimentation that exposes them to grave risks without the benefit of any direct therapeutic effect to themselves. Owen MacMullen, another Catholic theologian and an authority of the history and philosophy of science, and of Galileo in particular, recently wrote of the exegetical principle that plays such a crucial role in the Galileo affair. When scripture and science seem to conflict, one should depart from the literal reading of scripture only when the scientific alternative is clearly shown to be true. And it's appropriate, therefore, that we should now turn briefly to the science from which it is longer established, some of which is longer established and others of more recent origin. So here I speak of the contrasts in religious groups concerning when life starts and of the beginning of life, and the view taken by Aristotle and Aquinas and expressed by Pope Pius IX in 1869. If we look, first of all, at the two top parts of this slide, it reminds us that the removal of 4 or 8 cell egg from the uterus and separation into its constituent parts and placement of each into different recipient animals results in clones of normal calves. In other words, these embryonic cells at this stage are totipotent. Each has the capability to produce a fully formed embryo, a fetus, and a live young. Each totipotent cell is an individual in potency, and whether this is generally true in all species is less certain. Second, from the bottom 2 slides, the pictures that you can see, the blastocyst is divided into 2 equal parts through the thickened area on the left of the inner cell mass, which is the section that's going to produce the embryo, and that is done with a sharp diamond knife, and each half is placed into the uterus of different sheep, produce genetically identical offspring at the end of a normal pregnancy. In other words, this stage of early pregnancy, even at the inner cell mass stage, when the embryo is ontologically indeterminate in that it can form 2 individuals. Third, until about day 14, when cells pile up to form a primitive structure from which the brain and the spinal cord will emerge, embryos may divide to give rise to genetically identical or monozygotic twins, as we saw in Hurlbut's slide, but this is in a different orientation. After this time, identical twins rarely, if ever, form. And Wood and Trounston wrote in 2005 that the violation of an inherent uniqueness of an individual already exists in natural reproduction with the occurrence of monozygotic twins. And this slide shows different ways in which these twins may arise during the course of early pregnancy. In other words, whether there will be a single or a twin pregnancy is not certain. Whether there will be one or two individuals is not certain within the first 2 weeks. The fourth point I want to make is that there are now known to be key genes that are involved in maintaining embryonic cells in a pluripotent state. And as you can see on the left-hand side, here's a stem cell, which can give rise to different cell types, or it can self-renew, as indicated here. And these key genes include ones that code for proteins, such as one that is called nanog, which is fittingly named after the mythological Celtic land of the always young. In other words, the gene that is keeping these cells always in a young state and in a renewal state. Nanog is thought to be at the heart of this network here, which is shown on the right-hand side, a series of proteins which keep these cells in this pluripotent state. And when members of this module are lost or inactivated, these cells then start to lose their pluripotency. So the argument I'm wanting to draw here is the picture that emerges is that during the first 2 weeks of pregnancy, early embryonic cells gradually pass from a state of natural totipotency, where they can form individuals, to pluripotency, to multipotency, and ultimately to unipotency, to single cells themselves. And their development, the loss of their developmental potential during this period corresponds with a period when about 50% or more of human embryos die naturally during early pregnancy. That's due to serious chromosomal abnormalities, and it's especially numerous in humans. Now, this gradualist view of the development of the embryo is seen as consistent with the way that cultures react to early embryo loss, according to the House of Lords report in 2002, which said this: although would-be parents may feel sad at the natural loss of early embryos before implantation, there is no public mourning ritual associated with it, nor is there the loss of surplus embryos left over from IVF treatment. Now, such scientific data have illuminated this distinction for many of us between genetic individuality, which is associated with implantation, and the gradual emergence of personal individuality or individuation about the time of the appearance of the primitive streak, which is illustrated here at the bottom, around day 14 or so. And Oliver O'Donovan, the professor of moral and pastoral theology at Oxford, describes in his book Begotten or Made that the embryo is a suitable object of experiment because it is not like us in every important way. It has no personality, it is us but not us, it is undifferentiated humanity in which spirit has not yet risen out of matter. Personality has not yet emerged from its biological substrate. So when we address the matter of intentionality behind the use of embryonic stem cells, it's to learn more about ourselves and how to heal others. Now, among the early advocates of the distinction between genetic and personal identity was the distinguished geneticist Anne McLaren, who went on record as saying, if I had to point to a stage and say, this was when I began being me, I think it would have to be here, referring to the day 14, day 15 period of the appearance of the primitive streak. And Swiss bioethicist Alex Morans puts it rather more graphically when he writes that there is no automatic congruence between genetic identity established at fertilization and individuation established later. Nobody ever suggested, he says, that monozygotic twins should have a single passport or a single voting ballot. And from this viewpoint, the traditional insight over the centuries remains valid that a potential human individual cannot be an actual human individual. The new reality is that a blastocyst is both a potential child and a potential cure. Now, few would wish to deny research that could lead to new forms of treatment for heart disease, diabetes, Parkinson's, motor neurone disease, and so on. However, scientists who argue this case for gradualism and intentionality are frequently charged with a repugnant utilitarianism because embryonic stem cell research is morally unacceptable because of the destruction of human embryos. So, as we've seen from Bill, the ethical dilemma drives the search for alternatives, and I won't go through many of those which he's already referred to. Remarkably, the development of new ideas in this area is quite striking. In fact, the alternatives are becoming so numerous that nearly every week a new one hits the headlines. And a few weeks ago it looked as if, to me, as if this evening's discussion could actually become superfluous. Remarkably, a landmark experiment in Kyoto has shown that out of 25 likely genes, 4 are needed to change adult cells on this side of the slide back to those that resemble embryonic stem cells. The 4 are listed here: OCT4, SOX2, c-MYC, KLF4. And these reprogrammed cells going back in this direction, which, as Martin said at the beginning, is something that is certainly rarely, if ever, seen. These reprogrammed cells can contribute to almost all tissues in the experiments that have been carried out in Kyoto, just like conventional embryonic stem cells. If only similar results could be obtained with human adult cells and at a higher level of efficiency, much of the acrimony in this debate could dissolve, and the courageous work and argumentation of people like Bill Hurlbut and his colleagues would become redundant. However, deontological issues will remain about venturing onto slopes that are slippery, marketplace aggression which distorts the science through conflicts of interest and challenges to integrity, fraudulent claims, scientists who will be as gods because humankind always aspires to immortality or self-aggrandizement. Hence the importance of regulations and rules and guidelines for new technologies. But even so, confusion can arise through inconsistencies, so that in one country, experiments on human embryonic stem cells could land you in prison, but in another, it could gain you a Nobel Prize. So, if intentionality behind the preparation of embryonic stem cells has life-sparing objectives, it's unsurprising that 34 countries, representing 3.5 billion people, about half the world's population, have permissive or flexible policies on human embryonic stem cell research. Among the most restrictive are Austria, Ireland, Norway, Poland, Germany, Italy, and of course the USA. So to summarise, 3 brief points. Advances in science tell us that the beginning of life is more complex than we once believed. The ontological argument that a unique genome formed at fertilization underpins the nature of the individual person does not fit well with the holistic picture of human functioning, which depends on complex regulatory networks, metabolic functions, synaptic plasticity, and mental processes, rather than being reduced to a hierarchical structure with genes which sit at the top of a command structure. Secondly, the scientific findings point increasingly towards gradualism and a double aspect distinction between genetic individuality associated with fertilization and personal individuality emerging later. Nonetheless, those of us who advocate and support human embryonic stem cell research have to at least stop and contemplate the fact that every one of us was once an embryo exactly like the one they are destroying. The other extreme, those who wish to close down research should have the moral courage to sit face to face with a person recently diagnosed with Parkinson's and say, I am not willing to destroy a human embryo, even to save you and your family from the hell into which you are about to descend. Those sides which confront the pain of the new idea may find that the speed of progress in the biological sciences just could lead to a resolution of this ethical impasse. Thank you very much.
Well, I'm not a scientist, so you don't get any pictures. You've been sitting still for a long time, so I'll be fast. I learned about stem cells by being on the House of Lords Select Committee on Stem Cell Research, chaired by Bishop of Oxford. One of the things I learned was that there was no argument to be taken from the religious tradition. Some parts of the Christian tradition have followed Aristotle in their belief that 40 days, or 80 days, were landmark moments of moral status. Others have pushed it back to conception, and of course we've heard about the change, no longer seen as a change, but well known to be a change in the Roman Catholic position in the latter part of the 19th century. So I don't believe that arguments from authority will get us very far, whether we cite Genesis, Job, or encyclicals, or indeed contemporary Jewish, Christian, Hindu, Sikh, or other sources. We heard them, they disagree. We also see very strong evidence of disagreement among Roman Catholics in this country, some of whom regard IVF as a permissible thing, others of whom regard it as totally impermissible because inevitably it involves the destruction and the foreseen destruction of human embryos. So let's leave the argument from authority behind us. What can we say? Well, I believe this is a very unusual moment in our debates about ethical issues, in that it's rare for new science to raise new ethical issues. Usually, one's amazed and excited by the science, but when one thinks about it, one can see parallels. However, all of those religious positions and the secular positions, which are just as varied, have taken it that we can tell a story about human development from the less to the more differentiated. What I think is very interesting and challenging morally about the science that we have heard is that we can see that there is de-differentiation as well as differentiation, that there is a retreat from specialization. Now that has profound implications, because if there is a possibility of de-differentiation, and then of course perhaps redifferentiation in a new direction, we have to decide whether that de-differentiation leads us to alter our ethical views. Now, Bill made much of the thought that you don't de-differentiate to totipotency, but only to pluripotency. However, it is unclear at this point whether that is simply a matter of not de-differentiating to the point where you get the placenta as well. It seems to me that we better, on the whole, look this new science squarely in the face. If de-differentiation is possible, then we have to ask whether it should be done at all. It takes us back from a specialized cell to something different. Now at present, and this was the basis I think of Bill's presentation, that a reversal of specificity is done by taking an oocyte, and in the case of human cells, a human oocyte, or very much more controversially, one from another species. Now it seems to me that, although we have not discussed it, but that the harvesting, which is a ghastly word, but it is used, particularly in the US, of human eggs is a very serious medical intervention, and even with willing donors, not one that is going to be possible on a large scale. So these newer ways of getting a reversal in the way a cell works are of the utmost significance, because we cannot expect to build stem cell research on women donating large numbers of human eggs for research or for therapies. And there are, of course, different considerations about how many would be needed. But equally, I do not think we can assign special moral status to every cell in our bodies. If I rub my skin like that, I'm destroying quite a lot of cells. If those somatic cells, skin cells, could have been reversed to have the potentiality for any cell in the human body, then I think we have to acknowledge that the argument for potentiality is dead unless you wish to put the whole burden on those cells in the blastocyst which do not develop into somatic cells, those which develop into the placenta. Maybe it will look different in a few years, but I think at the moment it is looking as though if we believe in human dignity, which I do passionately, we better not hinge the arguments to the status of the human embryo. The human embryo is wonderful because of its potentiality, but just possibly other human cells are equally wonderful because of their potentiality. And if we want to talk about human dignity and moral standing, I think we need to talk about many other things, even about the soul. And to my mind, this whole fantasy that the status of the embryo, which is material, is decisive, involves a curious materialism borrowed from Aristotle by the religious tradition. We did have time to talk about Aristotle's very different doctrine of the soul, but I think we're in a new world, and I have never on any other issue thought that science took ethics in so radically new a direction. Thank you.
May I invite the speakers to join me on the platform, please? It's just been pointed out to me that I'm like this cork. I shall fall over the edge of the world in a minute. But can we? We haven't very much more time, I'm afraid, but I think they've been most stimulating and interesting presentations. Do we have anybody who wishes to raise any particular question, either to what I would regard as the panel or to an individual speaker? There are wandering microphones there. Please don't be shy. Here we are. Just over here, please.
Many thanks for stimulating talks. It seems to me that there's an argument that if in certain circumstances any human somatic cell can have the potential to form a new human being that we would all recognise as a human being, and that may not be possible now, but in principle, if I understood Baroness O'Neill correctly, that looks to be at least on the table, so that there's a form of potential that any somatic cell has, then that means the end for any basis of human specialisation and dignity based on potential, that it would lead to any cell having some special quality that we would need to respect, and this is proving too much and therefore can't prove anything. It seems to me that that's an interesting and important thing on the table. I'd be particularly interested to hear what Bill had to say about that, but also what Brian would see perhaps from just the feasibility of that scientifically.
Brian, he mentioned you.
That question sounded to me like one for Onora.
Brian, what's your opinion?
Well, I think it's interesting, Patrick, that you picked up a point which has been evident in the scientific realm for several years by now, right in fact from the time of the cloning of Dolly, where the use of a mammary cell which was transferred into an egg of another animal resulted in Dolly, and that demonstrated that in fact it is possible to reverse, to reprogram a cell which everyone thought was an adult cell and that was not possible to reprogram, that it is possible to do that in mammals, and this was the first demonstration. It followed on earlier work, of course, of John Gurdon, now here in Cambridge, but when he was in Oxford, in which he was interested in the question, when cells specialise, do they actually lose genes and therefore become specialised cells? And his experiment showed very clearly that that is not the case. So that each cell within the body has this potential, as you say, and as was highlighted by the Dolly experiment, and as has been picked up by the comments that Onora has made.
I would like to take on that analysis. By the way, I understood that Dennis O'Neill actually said the same thing. You pointed to the difference implied by the capacity to take any adult body cell and somehow convert it into an embryo. But in fact, that's not what's happening at all. You're taking the nucleus of a cell, which, as Professor Heap pointed out, is just a portion of the cell, and putting it into another environment. This does not involve the transformation of an adult body cell. It involves the creation of an entirely new entity. Now, we know that you can change things from one thing to another, but does not mean that the intrinsic powers of that thing was already there. I would say that there's a distinction here, that there's a difference between something having the active potential or potency of an embryo, in other words, intrinsic to itself, the powers of natural development. There's a difference between that and a passive potential from which an external manipulation could change it to become part of a system that had the equivalent powers. So it's a difference between active and passive potential, and I think it makes all the difference in the ontological world, if you will.
There may be arguments that could establish that point, though I don't think we've heard them this evening. Active and passive potentiality are very tricky concepts to keep apart, and I'm just not certain whether anything could be done along those lines. Talked with a lot of theologians who are pretty dubious about it. And of course there are people who are absolutely straight that we mustn't ever destroy any human embryo, and therefore no IVF at all. But, and I respect that position very much, but they, in my experience, have not tended to be people who are following the science very closely.
I won't just want to come back to that. I think there's something odd in this statement. You know very well if you take a body cell and put it into the womb, it's not going to grow to be an embryo. You know very well when you put an embryo in that it has the chance of doing that. The complex philosophy, active, passive, whatever you want to call it, it takes a dramatic external manipulation to make any part of a body cell participate in the formation of a new embryo. That is not the status of an existing embryo. It has within itself the intrinsic powers for its own development if it's a fully constituted embryo. And since we're on the subject of potential, intrinsic potential, innate potential, that define the thing as a thing of a specific kind. Let me respond to the argument on twinning. And I'd like to put a question to Professor Heap. If one accepts the argument from twinning, and I have to say that early on in my service in the President's Council, Brian and I were at a meeting together, and we had a conversation, and he told me about his thoughts on this and said that ultimately the argument from twinning was the most compelling and difficult issue for him. And I fully concur. I think my conversation with him was the best one I had in preparing for my task on the President's Council. In the end, I felt like it was not finally a compelling argument. So let me put a question to you. Even if your argument were true of, say, let's say maybe 30% of embryos can become twins by some, let's assume it's an innate power even, which I don't think it is. I don't think there's any evidence for that. I think it's a result of a disruption and restitution, as I said. But let's assume for a moment that it is an innate power in the thing created. The fact still is that 70% would then not have that, and therefore would they have a different ontologic state than the one that could twin?
Since we had our discussion a few years ago, I'm not content with simply isolating this twinning issue as the one that is persuasive one way or the other. I think it is one of several arguments which I try to draw together in terms of the scientific argument, and that it simply sits alongside these other points that I made concerning totipotency, the extent to which there are genes that are clearly changing in terms of maintaining pluripotency and directing cells down particular lines. So, although I still believe that the twinning argument is an extremely difficult one to circumvent, and it's certainly been one that's been very influential in my own thinking, I don't think it's by any means the only one that I would want to draw attention to.
It's not, if I could put in a comment as a mere biologist without great ethical roots, it's always been very interesting to me that the whole process of nuclear transfer or cloning was at one time anathema, and yet now it seems to me that partly because I think of the argument I'd like to ask you about this of individuation, genetic individuation and fertilization, that actually the nuclear transfer type of procedures are almost becoming favoured. Is that a reasonable?
My perception of the ethical debate in this country, my perception of the ethical debate in this country, which at the moment probably centres around proposal for further amendments to the Human Fertilisation and Embryology Act, is that that impression is partly because, and this may sound paradoxical to you, Bill, we actually have much stricter legislation than in the US. In the US, the legislation merely forbids federal funding of this work. It doesn't forbid the work. We regulate it extremely tightly. Anybody who embarked on reproductive cloning could serve rather a long prison sentence. So, for violating a license or proceeding out of license. That's why some of these sort of people who, I suppose, fasten on the grief of the childless and propose to conduct cloning experiments in this country have, I think, simply not read the legislation and the penalties they'd be open to. So I'm not sure that in this country there is much ease about the cell nuclear transfer. First of all, it's criminal if one were to implant the resulting embryos. Secondly, there is great unease about egg donation, and of course the sale of human gametes is again unlawful in this country.
It was just made lawful, I thought, wasn't it?
No, you can get your expenses. That's different.
There's no spot market. If anybody else would like to?
While you're standing up, let me affirm that that is indeed true, that in the United States, the law relates to federal funding, and except as regulated by the states, there is an amazing lack of laws governing this. That's the result, however, of a society which was completely unable to come to grips with the abortion and then subsequently the issue of IVF. And the reason that the federal government is holding the line on the current prohibitions against embryo-destructive research is because there's the argument that the whole society collectively should not endorse this. But it is true that it's perfectly legal with private funds. But that doesn't necessarily mean that it's more or less permissive. It depends on what you define as permissive, because at least from the perspective of those with moral objections, it does not involve the collective institutional endorsement of the instrumental use of human life. And that, well, at least could be argued if you have moral concerns to at least give the United States some grounds as a collective society expressing moral concerns on this.
Okay, we've got down 3 questions. One here and 2 over the back there.
Yeah, I was hoping that Onora and Neil could comment a little bit more on why you think the possibility of de-differentiation poses a problem for a picture of moral status as being attached to an organism. Sort of Bill's argument was really about attaching the moral status of the embryo to the fact that it has an organizing principle of life. It's got, sort of a developmental biology point of view. This thing is a thing that becomes a person, not the fact that it is potential, but rather that it actively strives and overcomes obstacles that prevent it from becoming an organism. So I just didn't quite understand how the fact that cells could potentially de-differentiate sort of poses a problem to that way of defining human dignity.
I think I'll probably end up repeating myself, but these terms, active and passive potential, are ones that I think are very difficult to tie down. Potential is capacity to become something. Now, it may be the case that ultimately we discover that the de-differentiation stops at a certain point, but the science is not looking as though it points in that direction. The consequence of adopting the potentiality argument is that one is exposing oneself to losing a grip on all the things that matter to us, including human dignity, because of, as it were, hitching it to the wrong materialistic point.
Thank you. Coming over here, please.
I just wondered that, as this is a Faraday Institute talk, not a series of discussion, it was Faraday was both a practicing Christian and a practicing scientist. The middle speaker dealt with the issues from a theological point of view. There was a lot of implied theology in the first speaker's talk, but without actually any specific theological references. How would they develop their arguments from a theological point of view, given that the audience here are Christian, and therefore looked at it from the point of view of a Christian worldview or scriptural worldview?
Do you wish to answer?
Sure.
You start.
Well, I was fashioned in the crucible of criticism over President Bush's Bioethics Council from the moment it was appointed, that we were going to just put forward religious arguments. So from the very beginning, I had my armor on that. And but I am a practicing Christian, and I think that everything I've said is consistent with my faith, but I don't think that it depends on it in any way that couldn't equally be argued from a non-Christian perspective. I mean, first of all, I agree with Brian that scriptures are not clear about it. The tradition is pretty clear. The Didache, the oldest document in Christian tradition other than the canonical writings, is very clearly against abortion. The Jewish tradition is the affirmation of life from its beginnings. I'm not comfortable with the claim that that Judaism accepts 40 days. Does that mean that 40 days you could use the embryo instrumentally up to that? You could gestate them and use them to then? Why not? If you could get an artificial endometrium. I don't think so. I had a long conversation with the chief rabbi of France on this and laid it all out. And in the end, he said, those principles, 40 days, the embryo is like water for 40 days. He said that has nothing to do with laboratory experiments. That's related to issues of penalty and purity. Nobody 2,500 years ago, when the Babylonian Talmud was being constructed, had any notion that one day we were going to create embryos in a dish. So I don't want to tell Jewish people what to think about their tradition, but I think the Jewish, Christian, and Muslim traditions are very much life-affirming traditions. And I think maybe it's time we rethought all that based on scientific argument. And I think the scientific arguments I put down are the part of the unbroken, seamless garment of truth that we ought to use, not purely theological arguments. However, I will say this, that some of the greatest scholars, Thomistic scholars and philosophers, maintain that had Aristotle and Aquinas after him known what is known by today's science, they would never have assigned it 40 days, and certainly not 40 and 90. I don't feel like the arguments that I made are actually religious arguments, as you say, they're compatible with religious arguments. And I honestly don't feel like the comments that have been made in criticism of my arguments are adequately rigorous so far in this debate. I think this gentleman who asked the question was asking a very real question. Isn't there a difference of kind between something that has within itself the intrinsic powers to develop, and isn't that a difference ontologically between that which could be made if something were added to it?
I would just sooner hinge human dignity to more significant things than developmental biology. I think it's too important to put it at risk this way.
I'm not giving away anything of dignity. It seems to me that those who give away the dignity of life in its earliest stages, and most of this life, at least currently, is unquestionably life with the potential for unfolding in the human trajectory of development. That seems to me to be the challenge to human dignity. If you give away life for one of its stages, what argument do you make to not give it away slightly? Do you really think that if it turns out that the most useful cells were 16 days, that the utilitarian argument couldn't find its balance of goods to move the line?
Well, look,
Yeah,
nobody has
step,
put
I
forward
might
a
say.
utilitarian argument here. You ascribe one to Mary Warnock, but nobody here, as to my ear, has put forward a single utilitarian
We are
argument.
running out of time. I think this is extremely good, but I did promise one extra question over there.
Thank you. My question actually hinges slightly more technically. Am I audible? Because I dislike microphones. Am I audible?
Yes.
Yeah. My question is slightly more technical with regards to some of the alternatives you were suggesting, sir. Now, as I understand it, you're suggesting a method of RNAi to suppress genes to prevent the formation of an embryo. Now, I would imagine, and I'm hoping you can possibly clarify, because I would assume that you're rather more familiar with this than I, that transferring from a mouse model to a human model, you are going to have less than a 100% success rate, at least in the initial stages. And a less than 100% success rate presumably means that you are going to create failure to disrupt genes, and therefore going to create actual embryos in the process. So my question is, how likely do you think that is going to be? And does that pose a moral problem? How many embryos being successfully created in the process? Is that not in some ways a utilitarian position in of itself?
Yeah, go ahead.
I would add a supplementary to that is, how do you know?
Well, it's like, first of all, that's an odd comment in an environment where I'm just trying to defend the embryo in the first place. I mean, if you give them away in 14 days, what's the difference? Well, I do. But I assume you don't agree with giving the embryos away in 14 days. You asked that question. So let me try to meet that. We are working with a very high-level group of moral philosophers and scientists, including people from a range of religious faiths, people who are familiar with biomedical science, and we're trying to find the answers to that question. The next step in our procedure, since it works perfectly in mice, the next step is to transfer those studies to non-human primates. Their rhesus macaques have a very similar endocrine system, and we'll have to evaluate how convincing the results of those studies will be before moving on to human cells. And in the final end, you do have to try something, but there's a difference between absolute certitude and moral certitude. We do that all the time with medicines. We develop them in animal models and we move on. Here we have a high bar, I think, very high bar to confidence. But I think it's possible that we can meet that with a great deal of certitude. So, and maybe something approximating absolute certitude, maybe absolute certitude, but without knowing. By the way, you don't need to just silence one gene. It may be possible to do 3 or 4 different manipulations simultaneously that preclude the totipotent potency of the thing. So we won't proceed unless we have high confidence.
Thank you very much. I think that you used a term now which might sort of wind us up. Absolute certainty, or absolute certitude, I think you said. I think that our real problem is that we, in the scientific sense, do not have absolute certainty. And I think one of the things that we're seeing in these moral debates is the clash of philosophies which would like to draw absolute lines for very good reasons, and human scientific endeavours, many of which do not draw absolute lines. And there is it is difficult to be certain all the time. I'm not going to ask you to reply to that, you can have more chances. But I believe, am I right, please, that there are some drinks downstairs?
Drinks and books.
There are some drinks available downstairs for further discussion right now. Sorry if we've gone on a bit long.